And then what kind of carbohydrates are important? Anybody ever kind of looked into different kinds of carbohydrates? Or what food sources are the best choices of food sources for carbohydrates?
Well, I know whole grains and like wheat breads and oats, even popcorn is a whole grain. Those are good carbs.
I love popcorn. Bread, pasta, okay, yes, whole grains, that's an important one, okay? You can write that one down. What other types of foods, though, besides whole grains, provide carbohydrates for us?
Plant sugars like glucose and fructose.
Yes, those are direct sources of simple carbohydrates. But what are the, so it's not just bread, right? Like we don't just eat bread for carbs. What other things can we eat for carbs?
Even vegetables have carbs.
Yes, yes. And a lot of people don't think that, or they think, oh, it's not a big part of the vegetable. Vegetables. I know people, and it worries me big time. They do not eat vegetables at all, at all. Their diet consists of like pizza type food, cheese, meat, and bread. And I'm like, are you okay? Like, you need to go to the doctor yesterday. So vegetables are a great source of carbs. And why are these a good source of carbs? Any thoughts?
Well, I'm sorry.
No, go ahead. You were going to say
they have no cholesterol. This is their plant-based, so there's no cholesterol, so that's good. And also, I mean, I guess like they're natural. I don't know.
Mm-hmm. What else? What other things are in vegetables, do you think?
Oh, well, like vitamins and minerals that we need.
So now we're getting at it. Carbohydrates are important. They all provide energy at the end of the day, but some carbohydrates are better than others. Okay, carbohydrates that also have other good things in them. So when you eat a vegetable, you're getting carbohydrates, but you're also getting minerals and vitamins. Okay, fiber. When we think about fruit, okay, if I want carbs, if I want glucose, I could drink a glass of orange juice, or I could eat an orange. Which one's better? Well, one has about half the amount of sugar and double the fiber. Okay, if you just eat an orange compared to a glass of orange. So it's important to think about what kind of carbohydrates. Okay, if I'm going to eat a dessert, I will literally argue this with my husband because his choice of dessert, he loves like Sour Patch Kids and Skittles. But if he doesn't like binge out on those, but like if he loves like sour candy. I don't. I would rather all day long eat a cookie, and I will argue this from a nutrition standpoint with him too. Like you are getting protein. Now a cookie's not great, it's loaded with sugar, but you're getting other nutrients in there versus just candy is like straight sugar and that's it. There's no other nutritional value, okay? So you want to pick a carbohydrate and you want to consider carbohydrates from a nutrition standpoint. You want to think, okay, what other things are packed in with these carbohydrates so that as I'm eating my carbs, I'm also getting other things with them. So that was just a simple little intro that I wanted to give as we think about carbs. And what we're going to do, this is not the only talk we're going to have about carbohydrates, but this is just going to get you familiar with what carbohydrates specifically are as a biological molecule, what they look like, how to draw them, how to name them. And then we're going to see later on how they're processed in the body and what our body does with those and all the different things that can happen, and then things that can go wrong with carbohydrates when they're not processed properly in the body. Robin could lead lecture on that day probably. That's where diabetes and things like that can happen. But today we're just going to talk about carbohydrates. So what is a carbohydrate? It's just a member of a large class of naturally occurring, and then here's the fancy words, polyhydroxyaldehydes and ketones. You know what this means. Poly means what? Many. Poly means many. Oh, Claire was about to say. Poly means many. Okay. So there's several, and then you all know what a hydroxy group is, an OH group. Okay, so there's several hydroxy groups. It's an aldehyde on a molecule that also has several hydroxy groups, OH groups. Okay, or it can be a ketone. Now, as a family name, the name ending is OSE. So that's why glucose, fructose, lactose, OSE, y'all are familiar with that. And you know that there's common names. We're going to use the common names for these carbs. And we're not going to learn 100 different carbs. We're just going to probably be familiarize ourselves with the top 5 or 6 that were that are common. But glucose, fructose, ribose is an example, and you can see the name ending OSE on each of those. So let's actually look a little bit more though. Different types of carbohydrates. You got monosaccharides, disaccharides, and polysaccharides. Okay. So a monosaccharide is what we think of when we use the word simple sugars. These are a single sugar unit. Okay. A mono meaning one. That's the kind of the word prefix for one. And then a saccharide is a word for a term for sugar. Okay. Simple sugar. So each monosaccharide contains anywhere from 3 to 7 carbon atoms. Okay, so they don't all just have like glucose has 6 carbon atoms, but some of them have 5. Some of them can have up to 7. That's, you don't hear that one as many. 5 and 6 is more common, but you can have anywhere from 3 to 7 carbon atoms in your monosaccharide. And then it contains either one aldehyde or one ketone functional group, just one. Now we have little differentiating names for these. Okay, we kind of combine, if it's a sugar with an aldehyde functional group, we call that an aldose. So we just take the family ending and we shove it together with the beginning of the word aldehyde so that we know it is a sugar with an aldehyde functional group. That's an aldose. If it has a ketone functional group, we'll call that, you'll hear me say it's a ketose. Specifically, we'll get even more, specific, and we'll communicate with this naming system how many carbons are also present. So for example, glucose has 6 carbons. So you'll actually hear people say it's an aldose. Okay, so what does each part mean? The aldo part means there's an aldehyde functional group in glucose. The hex part means there's 6 carbons in glucose, and the ose is telling us what the family ending name is, that it is sugar molecule. Okay, so you'll hear aldohexose or a ketopentose. Okay, what would a ketopentose be? Another example.
A ketone with 5 carbons.
Yes, it's got a ketone functional group. There are 5 carbons in that sugar, and it is the ose means it's a sugar molecule. Okay, so just get familiar with that language. This is a little figure from your textbook, just kind of outlining how we draw these, just so you get used to seeing them this way. So a monosaccharide is drawn like this. We always draw either the aldehyde or the ketone functional group towards the top. Remember, aldehydes are terminal. They're the very last carbon in the chain, or here they're the top carbon. So we'll draw that at the top, or the ketone would be carbon number 2, the next one down, because ketone functional groups are internal, right? They have an alkyl group branched off both sides of the carbonyl carbon. The green part that's highlighted is all the other carbons in the chain. And all these other carbons, so you'll have, any 3, 4, or 5 carbons in here, and all these other carbons will have different things branched off of them. Hydrogens and hydroxyl groups, okay? That's what you'll see, hydrogens and hydroxyl groups. And the number of carbons will vary just depending on what sugar it is, all right? But this is the common way we draw sugars in the straight chain form. Okay, and then always at the bottom is you have this other hydroxy group, CH2OH. You'll always see that at the bottom, okay? All right, so that's a monosaccharide. Disaccharides and polysaccharides, we also call these complex carbohydrates. A disaccharide, you just looking at the prefixes here, means we've got 2 monosaccharides linked together. Okay, so this is 2 mono linked together. Polysaccharide is just, let's just continue the chain. We just keep adding on monosaccharides, and we get these really long chains of sugars linked together. And those are used for our body in ways we'll get to later, energy storage and things like that. Okay, so that's some brief definitions. We're going to look a little bit more at how to draw these things. I'm going to get in briefly to a little bit of chirality, but I'm going to skip over. We're going to cover these next 2 slides, and we're going to skip a few slides. Okay, chirality and carbohydrates, and something known as Fisher projections. Fisher projections are just a way to draw sugars that help communicate chirality to us. Okay, So why is chirality important? Well, we know what a chiral molecule is. It's a molecule that has a specific spatial arrangement. Okay, chiral molecules can have isomers that are either mirror images, and they're not superimposable, or they can have isomers that are superimposable, and we have different names for these. I have a chart on the next slide that will help us remember what all the different names are and how they're different. But a chiral carbon, remember, it's a carbon bonded to 4 different groups, and that carbon has to be tetrahedral, right? And if it's bonded to 4 different groups, then it will be tetrahedral. Now, the 2 non-superimposable, meaning you can't lay them over one another, just like our hands. I can't lay my hands over one another and get them to perfectly line up. My pinkies and my thumbs won't line up. So if they're non-superimposable, but they're mirror images, meaning I could hold my hands across from mirror up to a mirror, and you get mirror images of your hands. So the 2 non-superimposable mirror image forms of a chiral compound are known as enantiomers. And we introduced this word back in, I think it was chapter 14. All right, but we have little letters that indicate the 2 enantiomers, okay, and how they're different. Okay, we have D and L. So what is right-handed, left-handed? Well, D, capital D and L, this gets confusing. Capital D and L, and then there's lowercase d and L. There's different properties with enantiomers that I briefly hit, and I didn't even put them on the exam back in chapter 14. Like some rotate light, they're just polarized light different ways, and that's not important. You know that. I'm just telling you they're different. But we have D and L, and in nature, sometimes you'll find that different forms are more popular in nature. So the D form of monosaccharides, because we can have chiral carbons in our monosaccharides, the D form is what our body uses and what nature uses. The L forms do exist, but for some reason they don't carry any significance biologically. Okay, so it is the D form of monosaccharides that are important to us, and I'll tell you in just a moment how the D and the L are different. Okay. Actually, we can look at this figure and we can see how they're different. Notice this is glyceraldehyde. It's a monosaccharide. Is it a ketose or an aldose? What do y'all think?
Is it an aldose?
An aldose. Got an aldehyde group there. So you see this little D and you see the L right here. Why are these here? How are they different? Here is how they are different. If you look at carbon number 2, so you see how the carbons are labeled 1, 2, 3? Carbon number 2 is a chiral carbon. So if you look at the 4 different groups bonded to carbon number 2, I'm just going to highlight them. We got a hydrogen, we got an OH group, we got a CH2OH, and then we have this aldehyde group. They're all different groups. So that is a chiral carbon. All right. So when you look at the chiral carbon in the middle, the hydroxyl group is branched to the right in the D form. Okay. But on the L form, the hydroxyl group is branched to the left. How do I remember this? L, left. Okay, L stands for left. So it is the D form of sugars with this OH group on the chiral carbon that is closest to this last carbon that is important biologically. Okay. Now, all right, many carbohydrates have more than 1 chiral carbon. This results in stereoisomers, spatial isomers that are not mirror images, so they're not enantiomers of each other. And these have a different name. They're known as diastereomers. I'm probably going to spell this wrong. diastereomers. Diastereomers. Okay. So enantiomers are mirror images of each other. Diastereomers are not. So how these words are getting, we have a lot of vocabulary words in our bank now when it comes to stereochemistry. And you might be like, how are these the same? How are these different? And I think this chart is really helpful. And again, in the folder in our Blackboard classroom, go to online classroom tab, there's a folder that says like extra info or stuff. There's a document in there that helps break down the different types of vocabulary type words for stereoisomers or stereochemistry. You can read over that to help you differentiate between all these things. So remember, we have constitutional structural isomers. Those are isomers who have a literal different connectivity. So look at the example here. See the fluorine atom highlighted in red? And in the bottom structure, the fluorine's on the very end. in the top structure, the fluorine is connected to the middle carbon. Different connectivity. Okay, those are structurally different. So we call those constitutional or structural isomers. Then you have stereoisomers. Stereoisomers have the same connectivity, but they have a different 3D or spatial arrangement. So we call them spatial isomers. That's the nickname for stereoisomers. Okay, so they're molecules that have the same connectivity. Over here, the structural, these have different. Meaning the atoms are not connected the same way. Okay, then we have diastereomers and enantiomers. Okay, diastereomers and enantiomers. All right, and enantiomers, these are mirror images. Okay, non-superimposable mirror images. Diastereomers, these are not mirror images. So for the enantiomers, imagine this dotted line right here through the middle is a mirror, okay? And you see how on either side, both things kind of line up, again, just like your hands would. Your hands are mirror images of each other. Diastereomers, these are molecules, for example, cis-trans isomers are, we didn't call them this back then, but I'm introducing this word now. Those are actually diastereomers, okay? They are isomers with a different 3D spatial arrangement, but the same connectivity amongst all the atoms, okay? They're just, things are pointing different directions in space. Then we really haven't got to conformers or rotamers, and I don't think you're gonna have to worry about that in this course, okay? But hopefully this little chart helps you space things out in your mind. It was a visual help for me, okay? All right, that's about as far as I wanna go with chirality. We will try to identify chiral carbons and monosaccharides, but we're gonna skip over, this and this practice problem. So I want you to jump down here to the Fisher projections. Yeah, I think that's the one.
Do you want us to just mark out all those?
Yeah, you can mark that out if it helps you. Okay. I briefly want to talk about this because again, you're going to see us draw sugars like this. These carbons will all be in a chain, and then you're going to see us branch all these groups off these carbons. Okay, and that's how we tend to draw monosaccharides in a straight chain form. And then we're going to learn today how to draw them in their cyclic form because they actually exist in ring structures. That's how they're most stable. So Fisher projections, these are structures that represent the chiral carbon atom as the intersection of two lines. Okay, what does that mean? We are representing a chiral carbon as the intersection of two lines. Okay, so horizontal lines represent bonds pointing out of the page, like towards you. And vertical lines, these represent bonds pointing away from you or into your screen or into your page. Okay, and then we always put the aldehyde or the ketone group towards the top or at the top. Okay, so for example, if we have this carbon right here, and it is a chiral carbon, and then you see what this is telling you, you see that the W and the X are actually bonds that point up, and the Y and the Z are bonds that point down. Well, if we press that flat on a page, it's hard for us to tell unless we have some rules here, which bonds point up and which bonds point back and so on and so forth. So this is just telling you the horizontal lines, these are the groups that are coming out of the page towards you. Anything connected vertically is pointing down into the page, okay? So that it matches up with how everything looks. So this is the actual Fisher projection. This is the way we would look at it on paper. It's two-dimensional to us, but this is actually representing a three-dimensional molecule. So these rules right here are helping us determine which bonds go up and which bonds go down to the page. All right. Now the wedges, these are bonds that point towards you. And the dotted lines or the dashed lines means that the group points away from you, right? So you can also indicate this with wedges and dashes. So this little picture is just showing you the same thing over here. vertical bonds point into the page, horizontal bonds point out of the page. All right. Okay. I'm not going to have you draw a lot of Fisher projections on the exam or anything. But there may be some problems on the homework and some things where this information will help you get the answer that you need. Okay. So D and L sugars, just a little bit more. And I think I mentioned this earlier, but this is kind of going to help you go back to it as you study for the exam. So I said this, in nature, most carbohydrates belong to the D family, all right? And in D sugars, this is kind of the official definition. The OH group on the chiral carbon that is farthest from the carbonyl group, it points to the right. So in the example that we looked at a few slides ago, where I highlighted that for you, there was only 3 carbons in our structure. There will be times where you have a carbon like glucose. I keep giving that example. Let's just say this is the aldehyde group up at the top. So what this is saying, in a D sugar, it says the OH group on the chiral carbon farthest from the carbonyl group. So I have 3 carbons, 1, 2, 3, in between my first and my last carbons. And if all of them have OH groups and if all of them are carbon, then what this is saying, the one farthest from the carbonyl group, well, that would be this one, carbon 3. That's the one farthest from the carbonyl group. So you always look at the next to the last carbon. Okay. Now you got to make sure that that carbon is chiral. If that carbon is not chiral, like it has 2 hydrogens bonded to it, then you go to the next carbon up. But usually the next to the last carbon will be your chiral carbon. And that's the one you say, okay, is my OH group on the right or the left? And that's how I classify it as D or L. Which leads us to the next definition. L sugars, the OH group on the chiral carbon, also furthest from the carbonyl group, points to the opposite direction, left, okay? And I just remember L and left. Why Fisher projections are important? This just helps us to identify D and L forms quickly, okay? Because we can literally, if this is the chiral carbon highlighted right here that we're interested in, we can treat this as a whole group. We can say that's group number 1, this is group number 2, group number 3, and number 4. And if we just draw this as a Fisher projection, okay, up on the slide above, they use the W, X, Y, and Z, okay? Again, I'm just using 1, 2, 3, and 4. We could simplify this, and this just helps us say, oh, this is a D sugar, okay? Because it just simplifies the whole molecule for me, and it helps us identify D and L forms quickly. Okay. You can see how L-allose is different, the OH group right here. So this carbon is chiral. It's got 4 different groups bonded to it. It's the farthest one from our aldehyde group up top. These other carbons in the middle are also chiral. It's just that we're looking at the next to the last carbon. That's the one farthest from the aldehyde group, okay? And when the OH group is on the left, that is L. When the OH group is on the right, that is D. When the OH group is on the left, that is L, okay? And it's the D sugars that are important to us biologically. But these are all enantiomers. Well, they're not all enantiomers. Some of them are diastereomers, but if they're mirror images of each other, they're enantiomers. Okay. I think both of these actually here, like D-glucose and L-glucose, are all enantiomers. D-allose and L-allose, those are enantiomers. Okay. Any questions on this slide? So I may ask you, what's the difference between enantiomers and diastereomers? Like on a multiple choice question on the exam, I could ask that or something like that. I want you to know that the D family of sugars are important and significant biologically. I want you to know how D and L are different. But this slide is where I want to spend some time today. Okay, so we can draw sugars in their straight chain form, and that is what you see right here. Okay, this is the Fisher projection form. Just why do we call it Fisher projection? Probably the person who came up with it, named it after them. But in nature, monosaccharides exist primarily in cyclic forms, okay? And not in straight chain forms. Now, we can obtain a lot of information from straight chain forms, but it's biologically significant that we draw them in their cyclic forms and assess how they behave and how they look and how they react in cyclic forms, okay? Oh, some familiar language. These hemiacetyls and hemiketals are represented by Haworth projections. So you don't have to necessarily know that it's called a Haworth projection, but that is the technical name for it. So we are going to learn how to draw these as Haworth projections in their cyclic form, okay? There's a few steps here. I encourage you to get a blank sheet of paper or have some space you can write for just a second, because I want you to walk through this with me. I'm going to explain it from the figure, and then we're going to do a practice, okay? I think, in fact, I have a practice in just a little bit. So how do we draw these from straight chain? How do we draw them into their cyclic forms? Step number 1, you're going to take the straight chain form, and look, all the carbons are labeled for you, 1, 2, 3, 4, 5, all the way down to 6. And then I want to point out the fact that there's 2 groups that are highlighted in different colors here. We have an oxygen on the aldehyde that's highlighted in blue or typed in blue. And then we have the OH group on carbon 5 that's in orange or yellow. So I want you to keep your eye on those 2 groups. Remember, we said that hemicetals and hemiketals can actually come together if you have an aldehyde or a ketone and you have a hydroxyl group, which is like an alcohol group in the same molecule, okay? And then they can come together and form a ring structure. And that's exactly what's going to happen here, okay? So step number 1 is we're just going to turn this on its side. So in your head, rotate this molecule 90 degrees. Or you could draw this out on your paper, rotated 90 degrees, okay? When you do that, we still have the same numbering for the carbons, okay? Carbon number 1 is still the carbonyl carbon in your aldehyde group. Carbon number 6 is now the one on the left. Carbon number 1 is the one on the right. So all we've done is we rotate this 90 degrees. Now I want you to notice another thing that happens when we do that is now all the OH groups are pointing up or down, okay, not right or left. If though the OH group was on the right, now it's pointing down, okay? If it was on the right, when you draw the straight chain form, now it's pointing down when we turn it 90 degrees. And if it was on the left in the straight chain form, now it's pointing up. We're always going to keep that type of orientation, okay, when you turn it, because that is going to match also how they point over here in their cyclic structure. And I'll come back to that in a minute. Okay, so the next thing we do, step number 2, it tells you coil the CH2OH group to the back. So that's carbon 6. So essentially, you're going to take carbon 6 and you're going to coil it around to the back. And that is what you see right here. We just swung it around to the back. Now carbon 6, you see the ring structure is starting to form. Now we just have to close it, okay? All right, so you're going to turn it on its side and then coil carbon 6 to the back. Step 3 is we're going to take this bond. Okay, so look at these, I'm going to highlight them. Look at these arrows right here, that one and that one. All it wants you to do is it wants you to bend carbon 6 up. So carbon 6 is laying flat and now we're just going to bend it up. Okay, so now it's right above carbon 5. Okay, so we just took this group, and then we just rotated it up right here. So now carbon 6 group, this is the same group right here. And you see what happens to this orange or yellow OH group when you do that? It swings up. So we turn it on its side, we coil carbon 6 around back, and now we just bend that last carbon-carbon bond up. Because remember, you can rotate carbon-carbon single bonds. It's perfectly valid to do this. So now we have this in perfect position to form a hemiacetal. We have this alcohol group here, and this is our aldehyde group on carbon 1. And this is how you form a hemiacetal. You have a reaction between an alcohol and either an aldehyde or a ketone. Here it's an aldehyde. So what happens is this hydrogen right here is going to see the oxygen in blue, and it's going to jump ship. It's going to bind over to that blue oxygen. Okay, so how can I better represent this maybe? this oxygen, this hydrogen right here is gonna come and it's gonna bond to that oxygen. And then you're gonna get a carbon-carbon or carbon-oxygen bond right here between carbon 1. Maybe I'll draw that in black. Between carbon 1 and this oxygen. You're gonna close the ring right here. Okay. Now again, you could go back and review the steps of hemiacetal formation to remind yourself, why does this happen? But we actually covered all of this in chapter 15. Okay, and that is how we end up with these bottom two. Okay, so we just close the ring. That's the last step. And then we have the cyclic ring forms. So we are going to practice drawing this today. That's what the lab is all about. We're going to practice drawing these, and then we're going to practice linking monosaccharides together as well. So we have two forms though, and I need to address why these forms are different. Well, in the name, how's the naming of these two forms different? The ones I just underlined.
One has an alpha and one has a beta.
One has alpha, one has beta, Robin. How is alpha and beta different visually? If you look at the rings, let me erase all my highlighting. How are they different visually? Do y'all see any difference? A lot of it's the same. There's only one thing that should be different.
The
Is it-
H and the OH are flipped.
On carbon 1, right here. Y'all see this? On a beta monosaccharide, that hydroxyl group on carbon 1 is pointing up above the ring. In an alpha monosaccharide, that hydroxyl group on carbon 1 is pointing down below the ring. Okay, now I know you're thinking, why the heck is this important? This actually has huge biological consequences. You want to know why these are important? The biological consequences. So for the beta, one is really digestible and one is not. Okay, so one, like cellulose, our body cannot digest cellulose. And I actually am blanking if it's the alpha or the beta form. Okay, I'll get back to you on that answer. But either the alpha or the beta form composes sugars like cellulose, these long polysaccharides, and they're not digestible by our body. Okay, they go straight through our body. Okay, there's certain parts of plants have a lot of cellulose. And then there's other forms, okay, like different polysaccharides like amylose, or it's called amylopectin. We'll actually get to those in a little bit, that are incredibly digestible and easy for our body to break down. So whether it's the alpha or the beta forms have really significant biological consequences on digestion and how our body handles those. All right, so definitely be familiar and be able to identify on the exam if this is an alpha monosaccharide or if it's a beta, okay? Or if I tell you to draw an alpha or draw a beta, know the difference between those, okay? Now, you also see this language right here. I want to point this out. It says, recall, the anomeric carbon is the one bonded to both the OH and the OR. So we've identified these carbons before, but that is carbon number 1 right here. Carbon number 1 has 2 oxygen bonds, one up right here to the OH, and then one back right here to the ether. So the CO-C bond, so that is definitely an ether group. It's a hemiacetal because you have an OH and then an OR group bonded to the same carbon. And we call this, as you can see right here, we call this the anomeric carbon. We call alpha and beta forms of a monosaccharide anomers of each other. Okay, so these would be considered anomers. Did I spell that right?
I don't know if you saw, Cameron had a question in the chat.
Oh, hold on 2 seconds, Cameron. A-N-A-M-E-R-S. A-N-O, sorry. These would be considered anomers. Okay, Cameron said, I thought the alpha beta meant different carbons. No. Alpha and beta just means that the hydroxyl group bonded to carbon 1 either points up or down. Okay, what you're talking about though is what we went over yesterday. Okay, so there's different uses of alpha and beta depending on what molecules we're talking about. What did we talk about with alpha and beta yesterday that Cameron's referring to? Anybody remember and can help us clarify?
Didn't it skip the actual first one?
Yes, this is when we were naming what? Yes, carboxylic acid, Cameron. I'm really glad you're clarifying this actually, because this might be confusing to a lot of people. When we were naming carboxylic acids, should be an OH, shouldn't it? Right there, okay? And then we have sometimes a long chain, okay? And maybe we can have different groups branched off like this. There's a IUPAC naming system and a common naming system. And in the common naming system, we recognize, I don't know what just happened. Robin, how? What did I just
Need
do?
to press the keyboard down right here on the right bottom. On the keyboard side.
Oh, okay.
Press
Okay.
that
Okay.
keyboard.
All right, we're back. Okay. Thank you.
There you go.
Okay, so we recognize where the carbonyl carbon is, and then they had a naming system. We call that the alpha carbon and the beta carbon, and we can even call that the gamma carbon. So yes, Cameron, that is for naming different carbons, but that is for when we are naming carboxylic acids or different molecules with those particular functional groups. Okay, there is a different way we use alpha and beta here, and that is for differentiating between anomeric forms of monosaccharides. I'm gonna say that again. There's another use for alpha and beta here, and that is for differentiating between anomeric forms. Anomeric means we've got two molecules that are anomers of each other. Because on the anomeric carbon, that is the carbon that's the hemiacetyl carbon or the hemiketyl carbon. The OH is either pointing up or down. I know this is very specific and this is very technical, okay? But there's two different uses for alpha and beta here. And you'll recognize it because you'll see alpha and beta either with a sugar molecule like glucose, fructose, hectose. You got to pay attention to the name ending. If it's OSE, then the use of alpha and beta is telling you where the OH group points on the anomeric carbon. If it's, how did we name carboxylic acids? Okay, it was something and then ic acid. If the name ending is some kind of carboxylic acid and you see alpha and beta in that word, then you know, okay, that is used to differentiate between my carbons over here. And it's just telling me what carbon maybe has what substituent bonded to it. We would call that beta hydroxy alpha blah 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 ic acid, okay, depending on what the molecule was. Does that help answer your question, Cameron?
I
Okay.
have
Can I
a
ask-
quick question.
Yes,
Um,
Claire?
so since the carboxylic acid, since those are going to be like the common name with using alpha and beta and gamma
Uh-huh.
for carbohydrates, like using beta and alpha, that's not a common name. That's how they're supposed to be named.
Glucose and fructose.
Yes.
Yeah, those are common names. We are not even gonna look into IUPAC naming for these.
Okay. Okay, good. I
And
was
we're
wondering-
only gonna stick to I have some tables down here. You are only gonna have to concern yourself with the sugars probably on those tables.
Okay.
And there's only 5 or 6.
Would
okay.
you say on the test it's going to be more common names?
Yeah.
Okay.
So we had a table yesterday. Let me pull that up actually. We had a table, where's chapter 17? Why is it?
Scroll
There
down.
it is. There it is. We had a table yesterday with a lot of common names, and I would really prefer that you familiarize yourself. So right here, okay, the names on here: formic, acetic, propanoic, butyric, steric, oxalic, those types of names, okay? Now, I still want you to know the IUPAC naming as well for these carboxylic acids, esters, and amides, but we are really starting to use common names a whole lot more, especially with monosaccharides and then disaccharides as well. We'll get to disaccharides in just a minute. Okay. So any other questions on the alpha beta stuff or naming? Okay, maybe you don't have one now. If you think of one, just stop me. So we've used all different kinds of words here. I want to explain really quick. I want to just review what is the difference between an enantiomer, and now I've introduced the word anomer to you. Okay. And an enantiomer tells you how the D and the L forms are related. They are mirror images, and we draw these in their straight chain forms. So enantiomers are mirror images. They're stereoisomers that are mirror images. We're looking at the straight chain, Fisher projection, and we're identifying whether the OH is on the right or the left. Okay. And then anomers, okay, are when you put them in their cyclic form. Did the OH group on the anomeric carbon, which is carbon number 1, this is the anomeric carbon. Did the OH end up above the ring or below the ring? Because we're going to see in just a minute, it's this carbon number 1 that we use to link monosaccharides together and form disaccharides and polysaccharides. So whether the OH group points up or down will have big indication or significance on how these things link together and how the long branch chains look. So that's how
I wish they
anomers,
were switched.
what is that, Robin?
I was just gonna say, I wish they were switched. Like, I wish the OH would be on the top in the alpha, because that would just make more sense to me. Like, alpha's on top, so OH would be on top. But now I just got to remember that they're flipped, and the alpha is actually in
Yeah.
the beta position. But it's okay.
Yep. Why can biology and biochemistry not just make perfect sense to us? That's a great.
Ooh, a good way to remember it is like alphabetical order, H comes before O. So
Oh, that's a good idea, Claire. Thank you.
now you're welcome,
H
Dave.
before O. Alphabetical. In the alphabet, H is higher up than O. So in an alpha, H comes first. H is on top. Thank you, Claire. I like that. Okay, I want to go back to red. So cyclic monosaccharides that differ on the positions of the substituents at carbon 1, as I wrote this earlier, but anomers. That's what this blank is. These are anomers. Okay, these alpha and beta forms of anomers, these are not enantiomers. That's why we have their own name for them, anomers. Okay. So let's review. Let's summarize everything we've covered so far. So far we've said monosaccharides. These are polyhydroxy molecules, and they have either one aldehyde or one ketone functional group. We said they can have 3 to 7 carbons. We've differentiated D and L enantiomers, okay, that they differ in the orientation of the OH group on the chiral carbon atom farthest from the carbonyl carbon. When we write these in Fisher projection, the D sugars have this hydroxyl group pointing to the right. The L sugars have this hydroxyl group pointing to the left. They commonly go from their straight chain, they form cyclic hemiacetals. These monosaccharides do. And conventionally, we represent them so that the OH groups on chiral carbons on the left in the Fisher projections point up. This is what I highlighted earlier. I said when you turn it on its side, notice that everything that was on the left points up. And I highlighted all the OH groups on the right, and I said those end up pointing down or below the ring. Okay, so those on the right in the Fisher projections will point down in the cyclic monosaccharides. And then we've talked about the hemiacetal carbon, okay, also known as the anomeric carbon. It's chiral, and in the alpha and beta anomer forms, the orientation of the OH is pointing down for alpha, up for beta. Okay, so that's what we've gone over so far. So this is just a review slide. Now we're going to practice this. Okay, so we have the open chain form of a sugar here. D-altrose is the name of it. And it has the the following structure. We want to draw it in its cyclic hemiacetal form. What's nice is that it's already been turned on its side for us. So here's what I'm going to do first, and here's what I encourage you to do. Number the carbons. 1, 2, 3, 4, 5, 6. And then I also go through on the OH groups, and I just make sure I'll write up and then I'll write down. I'll just do a D for down. Well, sorry, 6 is not going to matter here. 6 will have its Forget 6. Okay, I'll tell you why in a minute. So now, do you guys remember the steps? We've got it turned on its side. Now we're going to swing carbon 6 to the back. So here's what I want you to do. The way I will draw this, and the way, let me scroll up to the cyclic form. So like here, what you see, if we've got a 6-member carbon, then we need a hexagon, okay? And we always have the oxygen right here on the back corner, the back right corner, okay? So don't worry about all the OH groups and everything yet. You just draw a hexagon with the oxygen on the back corner. If it's a 5-member sugar, it might look slightly different. We'll practice that too. But for 6 carbon sugars, we're going to start with this base structure. So I'm going to draw my O. This is just how I go about it. Draw a line out pointing to the left. Now I'm going to bring both of these forward. And then I'm going to start bringing them together. And I'm going to draw a line out front. There's my hexagon. Don't worry about getting it like perfect, but try to structure it this way. Now we need to number our carbons. This O right here that I'm going to highlight, this oxygen is from the OH group on carbon 5. Now, how many carbons do we have represented in our structure right now? Remember, every time 2 lines come together, that's a carbon.
5.
We only have 5, but we should have 6. So always, we put the last carbon, this whole group, CH2OH. I'm just going to write it like this, CH2OH. We just point that up from carbon 5. So this is going to be carbon 6. I'll actually number these in black to make them stand out. Whoops, 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 that's wrong. That should be 5, and that should be 6. We always put carbon 6, and the atoms attached to carbon 6, those point up above the ring. So if we continue numbering, if that's carbon 5, that makes this carbon number 4, 3, 2, and 1. So now we have all of our carbons represented. Now we just need to put the OH groups on. Remember, in a line structure, you do not have to draw all the hydrogens, just the OH groups, okay? We'll worry about carbon 1 in a second, okay? Because carbon 1 is going to have an OH group, and we can have 2 forms, alpha or beta. All right, so we'll do carbon 1 last. So let's start with carbon 2 right here. We said this OH group needs to point up. So I just draw it like that, kind of up into the ring. Carbon 3 has an OH group that's going to point down. Carbon 4 down. Okay, carbon 5, I wrote down 2, but that's because I forgot. This is actually the oxygen, the OH group that's going to be used to form the hemiacetal. So I should not have labeled that as down. That one is the carbon. This is the chiral carbon farthest from the aldehyde, the carbonyl carbon. And so that is the one that's going to be used to make the hemiacetal. Okay, so now we could have 2 forms of this. We could have an alpha or beta form. If I draw it like this, did I draw the alpha or the beta form?
That would be alpha, wouldn't it?
Wouldn't it be beta?
The hydrogen would be pointing down. Remember, in alpha, H comes before O.
Yes.
As a
Okay.
place.
Okay.
Okay, so this is beta D. We could also draw the alpha form. Can I shrink this? Ah, I can. Okay, I'm liking this app better and better because I figured out. Okay, so if we also draw the alpha form, you draw your O with your hexagon. We always take the carbon 6 H2OH, point it up. And then we're going to draw the same OH up here. That's carbon 2. OH down on carbon 3, OH down on carbon 4. In the alpha form, the OH group will also point down on carbon 1. If you want, if it helps you to draw that hydrogen in on carbon 1, pointing up, then that is fine. There's our carbon number. So this would be the alpha D dress, okay?
Okay, quick question.
Yes.
So, like, if we were to see this on the exam, if we have to draw it, like, is it okay for us to draw it both ways, like, as the alpha and the beta form?
Right, so I would have to specify, if I tell you draw both forms, then yeah, you'll draw the alpha and the beta. If I want you to draw a cyclic monosaccharide, then I'll say, if I want it to be one form or the other, I'll tell you, draw the beta form or draw the alpha form or draw both forms.
Okay. I'll probably draw both forms just in case. On both of them.
All right. Okay, so that's drawing. We're going to do a lot of practice in that in the lab today. Now, some important monosaccharides. Okay, so these are, let's see, 1, 2, 3, 4, 5. Okay, so between these two slides, this is all one table, but I split it up into 2 sections. This is 5 common sugars, okay? And then here's a few properties of sugars. We know that sugars are sweet tasting, digestible, non-toxic, all right? Because there's all these OH groups, they're very polar molecules. They're loaded with these polar OH groups, and so they have very high melting and boiling points. We know what sugar looks like. It's a white crystalline solid. Definitely it's soluble in water. Okay, if you have a jug of sweet tea, my mom used to make sweet tea all the time at home growing up. I don't drink sweet tea anymore at all. But I remember she would dump the sugar in there and it just like immediately dissolve, right? We know it's soluble in aqueous solutions, and that means it's insoluble in non-polar solvents, okay? So I want to just introduce, you definitely need to know these 5, okay? D-glucose, D-galactose, I'm going to come back, hold on, D-fructose, ribose, and 2-deoxyribose, okay? Now I want to point something out. Right beneath the names that I underline, what do you see? It's an aldohexose. This is telling you it's an aldehyde or a keto sugar, telling you how many carbons is in it. Okay, we have both glucose and galactose are aldohexose. But then look here, you've got a ketohexose, so fructose has 6 carbons in it, but it is a keto group. So when this forms a ketal here, in this case, when it forms a hemiketal, it's actually only a 5-member ring. You see this? It only has 1, 2, 3, 4, and the oxygen makes the 5th place. All right. So this ring looks different than the rings up here. You got like a pentagonal versus a hexagonal ring. Okay. And what that means then is this is carbon 1 and this is carbon 6. That's the way we actually have 2 CH2OH groups pointing up above the ring here. Because if you draw this out, in a straight chain, the ketal group happens here. And then we're still the OH group on this 5 carbon. So these come together and form your ring. And an aldehyde, the carbonyl carbon is the very, very top one, but that's not what we have here. Okay. Ribose is an aldopentose, meaning there's actually only 5 carbons if you draw it out as a straight chain. And so when you wrap it together, it is an aldo group, okay, aldehyde functional group. It looks different. It only has an H right here. It doesn't have another CH2OH pointing up. That's because there's only 5 carbons and that aldehyde group is at the top. So when it forms a cyclic structure, you get what you see here in this picture. And then deoxy, or 2-deoxyribose, that is not labeled, is it? But what would it be? I believe it would have to be 1, 2, 3, 4, 5 sugars, so it'd be a pentose. And I believe it would have an aldehyde group on it. Yep. Okay, and then it just lists a bunch of different functions and uses, and so I don't want you to just straight memorize all of these things. But if I ask you, like, what is the common source of this sugar, like glucose? What's the common source of it? Okay. It tells you found in fruits, vegetables, corn syrup, honey. Okay. Galactose found in milk. All right. So galactose comes from animals, right? Glucose comes from plants. So I might ask you things like that. What's the common source of it? Or what's the common, the source and the purpose? Here's what I'd like you to know: the source and the purpose. Why is it significant to the body? Okay. So for glucose, stored as glycogen, that's the purpose of it. That's one purpose of it. And you don't have to know these things. Provides metabolic energy for mammals. So I might just highlight these. This is an important purpose for it. That's a second important purpose for glucose. Okay, I want you to know those things. Okay, and then it's sourced from plants. Galactose. So it's found in milk. It's also said that it's found in some plants. Okay, so galactose you could get from either, right? So the purpose of galactose, it's a component of brain, nervous system, tissues. It's metabolized to glucose for energy yielding pathways. Okay, don't worry about the whole deficiency thing. I think those things are important. Fructose, given from the name, it's in a lot of fruits, so it comes from plants. It's an intermediate in glucose metabolism. Okay, don't worry about how sweet it is. But there's the source and there's the purpose. Ribose, it is actually, I think our body makes a lot of it. So it's just internal. We source our own ribose. And then the purpose of it, it is part of our RNA, DNA, involved in protein synthesis. Just know that one. 2-deoxyribose, there's just one line there, part of DNA. Okay. So those are some important monosaccharides. Those are some things to know about them. What do I have next? Any questions on those so far? Oh, this is a really good practice. You guys, look at this. I want y'all to tell me the answer here. In the monosaccharide hemiacetal shown below, number all the carbon atoms, and then tell me which one is the anomeric carbon atom, and identify it as the alpha or beta anomer. So remember, the anomeric carbon atom, make sure it's chiral. It is the one that is part of the hemiacetal or the hemiketal. And in a hemiacetal or hemiketal, that carbon is bonded to 2 oxygens. I'll put a little note here. You're looking for the carbon bonded to 2 oxygens. That is going to be your anomeric carbon. We have to number them as well, though, and we're going to identify this as the alpha or the beta form. Anybody got the anomeric carbon identified yet? And Cameron says, is your comment in the chat for this, Cameron, did you say it's beta? Or is that for something earlier? Oh, that was earlier, okay. What do y'all think? Anybody? Y'all think this is alpha or beta?
It's beta, right?
So the OH is pointing up, which definitely means beta. So you're looking at this right here.
So you would label, oh,
So I keep wanting to say A and A, M-O-R, but A and O, M-E-R. This is the beta anomer.
so the
So which
beta,
one? Go ahead, Robin.
so the beta anomer would be labeled number 1, the first carbon.
Right beneath it. Is this carbon 1? This is definitely the anomeric carbon.
Is it?
Okay. If this is carbon 1, then what is that carbon?
Ooh. You're right.
We've got to number these all in a chain, like in a straight order. So like we can't go down on one bond and then go up and over to another bond. So this cannot be carbon 1.
So the anomeric carbon is not always carbon 1. then.
Not always carbon 1. It could be carbon 2, depending if you have an aldo or a keto monosaccharide, like a monosaccharide with an aldehyde group. Or you will often find. Let me give you a rule of thumb here. For an aldo, I guess you say os. For an aldose, meaning a sugar with an aldehyde group, ANO. I cannot spell today, everybody. A-N-O-M-E-R. The anomeric carbon equals carbon 1. For ketose, meaning a sugar with a keto functional group, anomeric carbon equals carbon 2. I promise as you practice drawing these, you'll see that happen again and again. So this must be some kind of keto carbon. So that's carbon 2. This has got to be carbon 1. So if we keep numbering from there, this would be carbon 3, 4, 5, and 6. Now you see how they're all in an order and we don't have to backtrack on any bonds as we number. Okay, so we identified the anomeric carbon and we identified it as the alpha and beta, or the beta anomer, and we numbered all the carbons. All right. Identify the carbocarbons in alpha D fructose, ribose, and then so we're just going to do one for time's sake. Actually, you know what? If we have time at the end, I'll come back to this problem. I'm not saying it's not important, but we really got to get to glycosidic leakages. Okay. Reactions with monosaccharides. So monosaccharides don't just sit static in our body. They react. They do a lot of things. We break them down to obtain energy. So real quick, recall aldehydes can be oxidized to carboxylic acid. Do you guys remember this? Aldehydes can be oxidized to carboxylic acid. So when we were talking about alcohols and we started really talking about oxidation reduction, we said primary alcohols, you did this yesterday on the lab too. Primary alcohols are oxidized to aldehydes via mild oxidation, but then those aldehydes can be further oxidized to carboxylic acids, and that's strong oxidation. So that's what we're just reminding ourselves here. Aldehydes can be oxidized to carboxylic acids. Now for sugars, we've got a lot of them have these aldehyde functional groups. So a lot of them, just like we know aldehydes can do, they can be oxidized to carboxylic acids. This can only happen though with the open chain form, okay? Not with the cyclic form, okay? So there's always in our bodies in solution, we have cyclic form and we have straight chain form, okay? And there's an equilibrium. This is reversible. We have a certain number of cyclic form and then aldehyde form, and they're just kind of going back and forth between one another if you have monosaccharides. When they're linked together in disaccharides or polysaccharides, it's not always the case. Okay, but when we have the open chain form, some kind of oxidizing agent, oxygen or other things, we know we have oxygen in our blood as well, along with our sugars, can oxidize these sugars, okay? So this is the aldehyde group. I'm gonna highlight it right here, and it can be oxidized to this right here, carboxylic acid. And we said yesterday that carboxylic acids at physiological pH, they have always lost their acidic hydrogen. So that's why you see the carboxylate anion form here. Remember, this is how we'll always look at pH 7.4. They will always have this negative charge. But that is now a carboxyl group. We call carbohydrates that can participate in this, we call these reducing sugars. Okay, they themselves are being oxidized, but they are reducing the oxidizing agent. So this is another part of chemistry that I don't love it because in my mind, it just confuses me. I would think you would call these oxidized sugars. That would make more sense to me. But I didn't come up with the rules. So we call these reducing sugars because of what they are doing to the oxidizing agent. They are reducing the oxidizing agent, but reducing sugars themselves are oxidized. But they are forcing reduction on the oxidizing agent that they are reacting or interacting with. Okay. Now, again, the main point I want to stress here is this can only happen, you can only have a reducing sugar if it has some sort of aldehyde group, okay? And you're in your straight chain form. Okay. I'm not gonna forget that. We're gonna move on. Okay, keto sugars can also be reducing sugars, only in basic solution, but we're gonna move on. Okay. Oh, this is really cool. So any of you, anybody know somebody who's like on the keto diet, like ketogenic diet? Any of y'all tried keto diets or know somebody that has? We all know what the keto diet is, right? Like you cut out sugars from your diet. Okay, now for me, that would be really hard because I love like desserts and sweets and things like that. But I picked them wisely, okay? I picked a cookie over a pack of Sour Patch Kids. But anyways, people who are on these keto diets, sometimes they just crave sugar. So we have a friend actually, he's been trying to lose a lot of weight and he's doing a good job. But he's had to adjust his diet. So when he really wants something sweet, have y'all seen those stuff in the store? It's like, they're like little peanut butter cups or desserts, but they're like no sugar. Okay. That's because they can say they're no sugar because technically they don't have like the sucrose and glucose types of sugars. They have sugar alcohols, okay, which are sweeteners. They're just not metabolized in our body the same way. So technically they don't affect our blood glucose levels, right? So they can literally say they're sugar-free, okay? It's sugar-free desserts. This is why sugar alcohols are important to us from a nutrition standpoint or a dietary standpoint. But this happens, so how do we get sugar alcohol? So in reactions with reducing agents, the carbonyl group is converted to an alcohol. So we just talked about how sugars are oxidized and they form these carboxylate anions. Now we're gonna see what happens when sugars are reduced, okay? So basically what we're doing here is we're looking at both directions. If we have an aldehyde, it can be oxidized. So this is the aldehyde. We know aldehydes can be oxidized to carboxylic acids, all right? On the flip side, the opposite of this would be reduction. They can be reduced to what? Anybody remember what aldehydes are reduced to?
Is it a ketone?
No, Cameron said it, primary alcohols. Okay, so we'll have an OH here instead, and then we still have this H, all right? So one way they're oxidized to carboxylic acids, and then they're reduced to primary alcohols. Okay, so that's what we're looking at here. The previous slide, we just talked about oxidation. Now we're going to talk about the opposite, reduction. So with reducing agents, the carbonyl group is converted to an alcohol. Okay, so now that's how we get sugar alcohols. Okay, so sugar alcohols, we have different names for them. So in fact, if you look at a nutrition label, I did this the other day. So my mom, she'll buy these popsicles for our kids. And I'm always like trying to watch the sugar content for my kids, you know, because grandparents come along and they just want to like shove cookies down your kids' throats. And I was like, Mom, how much sugar in these popsicles? Because usually popsicles just have tons of sugar. She's like, there's only 2 grams of sugar. And so I literally went to the store, I was there, and I was like, I'm gonna look at these popsicles, and I found the kind that she gives my kids. And I looked and it said sorbitol. Okay, anybody ever read sorbitol on a nutrition label? Okay. So what that means, this is alcohol, this is a sugar alcohol, okay? Sugar alcohols are named as derivatives of sugars, and instead of OSC, like glucose, sucrose, fructose, they have the name ending ITOL. So when I read that, I knew, oh, this is a sugar alcohol. Okay, now why would that worry me? Anybody read the next line?
Hard for them to digest the sugar alcohols if you eat so much.
If we eat a lot of them, okay, it can lead to unwanted effects, okay? It can have bad effects on your body if you consume a lot of these. So people who are on like a sugar-free diet, they have really got to be careful with the like sugar-free desserts they consume, okay? But this is what sugar alcohols look like. So here's the aldehyde group. When it's reduced, we know we're increasing the number of carbon hydrogen bonds when we reduce something. Here's the primary alcohol, okay? Remember, we have this palladium, or we can have a platinum or some kind of metal catalyst will help that, all right? Now, I put this next slide on here because it's hilarious. Somebody introduced this to me. Have y'all ever heard of the Harborough sugar-free gummy bears? Has anybody actually read this review before?
Yeah, I went through and I read them one day because we were talking about it in my food science class.
Oh my gosh, if you have not read this, just look at the title. Luke, whoever you are, he had a very bad experience with, and he details the whole experience. Don't take the time to read now. He says, "See you in hell, Harborough sugar-free gummy bears." Okay, so it's hilarious. I think beneath this, so many people said that this review is helpful. But he details his experience with sugar-free gummy bears. And this is a for real review, straight off Amazon. I just screenshotted this. Oh, down here. Look, I thought this was funny. Over 13,000 people found this helpful. So anyways, that's sugar alcohols, okay? Sugar alcohols are used a whole lot in industry for sugar-free desserts, but we get them by reducing aldehydes, okay, aldose sugars, all right? Okay, a few more reactions. So now what we're gonna start doing is some other reactions we can get with monosaccharides. Hemiacetals react with alcohols to form acetals. Okay, this is also part of the lab from yesterday, formation and hydrolysis of acetals, all right? So what is an acetal or an acetal? Some people say that way. It is just a compound with a carbon bonded to two ether type groups, okay? OR groups. So we have the hemiacetal and we react it with an alcohol. We have to have an acid catalyst to make this happen. What we end up with is an acetal. And this carbon right here, which was the hemiacetal carbon, it was bonded to an OH group and an OR group, okay? But one more reaction with alcohol and we get two OR groups, okay? It's showing you this alkyl group, this carbon group, it kind of just replaces this hydrogen. So they just kind of switch, they just substitute for one another and you end up with an acetal. And then you get water as a byproduct, okay? Water as a byproduct. Okay, so why is this important? Why is that? Why am I bringing that reaction back up? Because what we're about to start doing is understanding how monosaccharides link together to give us disaccharides and polysaccharides. And this is the reaction that's going to undergo. We're going to form acetals, okay? Disaccharides, polysaccharides, these are acetals that we get. So when cyclic hemiacetals, such as glucose or other monosaccharides, those are cyclic hemiacetals. When they react with alcohols, and guess what? Glucose, fructose, sucrose, these are alcohol, these have all kinds of alcohol functional groups, all right? We call the product glycosides, all right? So a glycoside is another word for like a disaccharide. So formation of a glycoside or like a disaccharide. We're actually not going to look at two cyclic sugars coming together. I'm just going to give you a very, very simplified form. Let's say we have glucose reacting with methanol, okay? This is just the simplest alcohol we could get. This is just a real example of what the figure just above shows you. Here, is the hemiacetal carbon. So this hydrogen that I'm going to highlight is going to come off, and then the CH3 that's in blue there is just going to substitute in its spot. And that hydrogen is going to join up with the rest of methanol, which is the OH part, and that's what makes water. okay, this CH3 group is what's gonna bond in place of hydrogen. And now you have an acetal. And this bond right here, this is the glycosidic bond, okay? Between the anomeric carbon and then oxygen. Okay. Now let's look at how we make disaccharides. It's the same reaction we just talked about on this previous slide. Now we're just gonna do it with 2 monosaccharides. We're gonna bring 2 monosaccharides together. We're gonna form a glycoside, but it's gonna have that acetyl bond. It's gonna be an acetal. So here is I'm gonna highlight on one, this first monosaccharide, cyclic monosaccharide, that's the hemiacetal. Again, I'm gonna point out this is the hemiacetal carbon. And this is an alcohol group. So when those react together, instead of CH3 like you saw up here, now we just have this whole group. Okay, it looks a lot more complex. So what's going to happen is this hydrogen comes off. Actually, it shows you in red. Let me erase this too. In red, so I'm going to change this. This whole group and this hydrogen, that's what forms your water byproduct. And then blue. All of this right here, see how this is the leftover part of that alcohol group? This is just going to slide over and bond right here. So what we end up getting is a bond that looks like this. This is the glycosidic bond. So the OH leaves from carbon 1, and then the hydrogen leaves from the second monosaccharide. And now you have this is like an ether, right? This is technically an ether functional group, but that is where the glycosidic bond, this is how monosaccharides link together. And you do this again and again and again, you get these long polysaccharides. Okay. Now, the way you see it right here, we have in this reaction, is this an alpha or a beta anomer? This one right here.
Would it be an alpha?
Mm-hmm. So when you have an alpha anomer, we're going to learn how to name these glycosidic bonds because they can look all kinds of ways depending on if you're using alpha or beta anomers. Okay. We would call this an alpha, and then we're gonna name the carbons that we're joining together. So we would call this bond, we'd say these 2 monosaccharides are linked together by an alpha, and then this is carbon 1, and this was carbon 4. So we call it alpha 1, 4. So that's gonna, if you say, okay, if you read something like glucose and fructose or some kind of monosaccharides are linked together by an alpha 1, 4 glycosidic bond, that's telling you exactly how they're linked together, which carbons come together, which alcohol groups are involved. Because there's all these other alcohol groups. Why not these? Okay. Well, these bonds tell you which alcohol groups from which carbons are involved. Okay. So that you can draw it properly. All right. Now, last thing. So that's formation of a glycosidic bond. Because water is a byproduct, you saw this yesterday. You can do this with monosaccharides too. You can take water and you can hydrolyze the bonds and break them apart. So the opposite of formation, we call hydrolysis. Okay, so hydrolysis of a disaccharide is simply breaking it apart into monosaccharides. And this is important in the body. We have to do this in the body. Let's say that we're not eating enough, we're not getting enough carbs in our diet and our blood sugar levels drop. Anybody ever had your blood sugar level just like drop out of nowhere? How do you feel when that happens?
Terrible.
Yeah, all of a sudden you're like shaking and you get tired, you can get dizzy, you can get nauseous and weak. So our bodies have to do this. They're supposed to regulate our blood sugar, and we keep a store of glucose in a different form in these polysaccharides. And at any time, it can start chopping off individual sugars and sending them into the blood to keep our blood sugar levels normal. Okay, so hydrolysis is really important. We have enzymes that help with this. But hydrolysis is really, really important when we need to raise our blood sugar levels in our body if we're not doing that enough through our diet. So what happens is the water is breaking these two apart. And so we have H and we have OH, that's water. So the H is, this is going to break apart right here. Going to break that bond. And the H is going to come over and bond to this oxygen and restore the OH group. And then this alcohol group is going to come over and bond to this carbon and restore the hemiacetal. And now we have two individual monosaccharides broken apart. Okay, so definitely for exam 2 on Monday, I want you to be able to walk through formation of a glycosidic bond and hydrolysis. I'm not going to ask you to draw all of this out, but if I showed you this, or if I ask you a multiple choice style question about, like what byproduct do you get in formation of a glycoside, or what molecule is used to hydrolyze a disaccharide, I want you to tell me water is involved there. Or if I told you, if I gave you a picture of a disaccharide, I said, okay, what kind of bond is this? We're going to look at some other kinds, but I want you to be able to determine alpha 1,4 or beta 1,4 or things like that. Okay, so let's do some practice with this. Draw the structure of the alpha and beta anomers that result from the reaction of methanol and ribose. Are these compounds acetals or hemiacetals? Okay, so we know methanol is CH3OH, but we need to go back up and look at our tables to figure out what ribose is. So we had that on our tables. I'm going to scroll up, sorry. Ribose, here's ribose. So we have this 5-member ring with the CH2, okay, and the OH groups are down, down, down. Okay, I'm going to remember that. Down, so let's go draw ribose. So I'm going to draw it right down here. So we got the O, and then we just kind of have like a pentagon. And then we had OH groups down, down, and down. And then right here, we had CH2OH. Okay, so draw this on your paper. This is ribose. And this is alpha. And then we want to react this with methanol. So try to draw the product here. You should draw the same sugar overall, but we're going to change one thing. And then we'll have OH. So what is going to happen right here? So this OH and this H are going to come off, form water. And the rest of the alcohol, you might have to turn it around, okay, is going to come over here and bond with this carbon. So what we should get is OCH3. So this was the part that was from methanol. Plus we get what's the other product? Plus water. So do we have An acetal or a hemiacetal? Or sorry, yeah, acetal or hemiacetal. So you're looking at, to decide this, you're looking at this carbon right here. That's the one bonded to 2 oxygen groups. Do I have an alcohol? So remember that, let me remind you, and for hemiacetal, the carbon has an OH group and it has an OR group. For an acetal, we have a carbon with 2 OR groups. So what do y'all think this carbon is, or this molecule is?
An acetal?
Mm-hmm. You might hear some people say acetal or acetal. Okay. Question though, we just said, this would be a gray open response on the exam. It's 10:30 though. I'm looking at the time. Actually just passed. So we're gonna pause here. Let me see how many slides, 9 slides left. We'll finish these when we come back. Let's come back. Let's come back at 11. Let's take a good little break. Let's come back at 11 and we'll finish these and we'll work on the lab, okay? All right, if there's any questions right now, Cameron, I'm sorry. Did I ever answer your question when you said, what's the difference again? Was that about a hemiacetal versus acetal? Okay, great. All right, well, if there's any questions right now, I'll stick around, but if not, I'll see you guys at 11.
Hey, okay, I do have a question.
Yes, Abby.
So I don't know why, but the reactions are so hard for me. Like I'm struggling on them. For the most part, do we try to count on like putting in an H2O or taking out an H2O? Does that make sense?
Like when you're doing hydrolysis and formation?
Yes.
Yeah, let me erase all my stuff. So let me try to simplify this. When you're forming a glycosidic bond, see the parts in red here? That's what leaves, and then the rest of them just come together.
Wait, did you write on your screen?
Oh, yeah, you can.
Sorry.
I always forget.
No, you're good.
Okay, so when you're forming a glycosidic bond, these parts in red, that's what leaves. And you see how that's water,
Mm.
HOH. And then we get a bond directly between. So this oxygen right here is just gonna bond right there to that carbon.
Okay.
And that's exactly what this is. And this is the new bond that we just formed between this oxygen carbon. So that's what we call the glycosidic bond, because that's what put the 2 monosaccharides together.
Sorry, Cassidy just got here. Okay, that makes more sense.
Oh, hi, Cassidy. And then you need to let her in, go for it.
You want to come over? Sorry, I was asking her a question about that. What we just did.
And then right here, Abby, so when we hydrolyze, we're gonna break the glycosidic bond. So we're breaking this one. And this already has an oxygen on it. This carbon right here now only has 3 bonds. Let me highlight those. 1 there, 1 there, and 1 there. We know carbon's on that side. So are we putting a hydrogen back on this carbon, or are we putting an OH group back on this carbon?
It would be hydrogen. No.
Well, the oxygen,
Oh, so
this
the oxygen gets
goes
the
with that
hydrogen.
molecule. So when we break
Okay.
this, so yeah, this OH has got to go back over to this carbon,
Okay.
and this hydrogen is just going to rebond to that. They both get OH groups in the end.
Okay. Awesome. Okay. Thank you.
Very welcome. Robin, did you have a question?
No, I'll always just stay just in case anybody else does so I can listen.
You bet. All right, see you in a bit.
All right, bye.